Hello everybody and welcome to the Pharma webinar DYNASAN 118, a multifunctional and versatile excipient. May I shortly introduce myself? My name is Michael Plum and I'm working in the product development and technical market support for more than 30 years. In today's IOI webinar, I would like to provide detailed information on the properties and various application options of DYNASAN 118, like solid dosage forms, hot melt coating, and more. So if you are ready, I would like to begin with the webinar. Let's start with the basic properties of the excipient DYNASAN 118. This is a high purity monoacid stearic acid is used, glycerol tristearate. So it's a 100% triester. It is based on natural and sustainable raw materials, and the melting point is around 73 degrees. The excipient has a neutral odor and taste, and it is delivered as a microfine powder. And it fulfills the USP/NF requirements for the glycerol tristearate monograph. But there's one important item to consider. This excipient tends to polymorphism. So what is polymorphism? A fat is based on fat crystals, and these fat crystals occur in several different states. Directly after production of DYNASAN 118, we have the so-called beta prime state, which is thermodynamically speaking an unstable state. And as you can see, the fat crystals are partially ordered in a grid with a so-called orthorhombic structure. But over a short time period, there is a state transition from beta prime state to the so-called beta state. This is the thermodynamically speaking most stable state, but this causes some effects like extreme volume expansion and the occurrence of white powdery particles. And as you can see now, the fat crystals are highly ordered in a grid with so-called triclinic structure. To visualize the effect, I have taken some pictures, starting with the beta prime state, which occurs directly after congealing of a molten DYNASAN 118. And if you leave this some days at room temperature, you can see the enormous effect. After transition to the beta state, there is an extreme volume expansion. It's like an explosion. You have a powdery and white particles. This, of course, has to be avoided because, as you can imagine, this would definitely destroy the packaging in which DYNASAN 118 is delivered. But there is an option to avoid this by performing a so-called curing step, which means a specific temperature treatment over a certain time period. Again, we start with the beta prime state directly after congealing, and after occurring of the beta state, after performing this curing step, you can optically see nothing. Nothing changes. It looks like at the beginning. You don't face this volume expansion and white particles, but indeed there has been a state transition to the stable and final beta state. So you have to consider this if you or your customer process DYNASAN 118 and where within this process the excipient is molten. Besides the fact that we are talking about a completely saturated ester, we have here additionally extremely low water content of around 0.03% on average. And this means that this excipient is extremely stable to oxidation. In the following picture, you can see a so-called Rancimat curve. A Rancimat is a forced oxidation process, applying high temperatures, in this case 100 degrees, and a constant airflow through the molten and heated substance with 19 liters per minute. And as you can see, if you look at the blue DYNASAN 118 curve, this is not a real curve, it's more or less a flat line. So you can see it's extremely stable compared to a mono and diglyceride like our Invita 900K, for instance. Poorly soluble drugs. This is one of the biggest challenges for the pharmaceutical industry today, and this is enabling the delivery of difficult to solubilize molecules like APIs, active pharmaceutical ingredients. So the solubility is an essential physicochemical characteristic of APIs, as it is directly related to bioavailability of the molecule inside the human body. This challenge increases as many of the difficult to solubilize APIs additionally show a low permeability. The solution to get rid of these problems is apply of specific techniques for manufacture of suitable application dosage forms in order to provide a satisfying API dissolution profile. Let's start with some application examples where DYNASAN 118 is successfully used. Start with the first application example, tablet production. Normally, standard excipients being used for tablet productions are cellulose, which act as a disintegrant or bulking agent, and then starch, talcum, and magnesium stearate, which act as a lubricant. These excipients are cheap, well-established and functional, but there is always a constant danger of possible tablet fracture. If you use DYNASAN 118 in low concentrations of 0.5 to 1%, this exhibits the following advantages. An improvement of lubricant effect. You will see increase of tablet fracture stability, and there is a disintegration acceleration of the API. You can see improve of flow properties of the tablet ingredients, of the other tablet ingredients. And last but not least, there is an option to generate a retarding effect by using higher concentrations of DYNASAN 118. Higher concentrations means 2 to 5%. And this increases the hydrophobic property due to the longer chain length, because the longer the chain is, the higher is the hydrophobic effect. And at the end, this means retarding. Let's come to the second application example, hot melt extrusion, or HME. This is a novel technique that is generating physically stable and processable amorphous forms of APIs. Because relative to crystalline APIs, amorphous solid dispersions improve bioavailability in more than 80% of cases, which is quite a lot. The mechanism of HME is to disperse APIs in a polymer or lipid matrix on a molecular level to form solid dispersions or solid solutions, which result in the processing of extrudates into final dosage forms, such as capsules, stick packs, or biodegradable implants. The HME process is continuous, which is important in respect to its reproducibility. It's a completely solvent-free and product-sparing process, as the process times are very short, only around 4 minutes. And within this process, DYNASAN 118 acts as a protective matrix for the drug molecule. Let's have a closer look into the process. It all starts with the API and the molten lipid, in this case DYNASAN 118. And then it enters the equipment, which is a so-called twin screw extruder. So you have rotating screws. Within this apparatus, you have a melting zone, a mixing section, and a homogeneous discharge section. And it ends with the cooling and pelletizing, and at the end, you will receive extrudates. And these extrudates are used in tablets, stick packs, or implants. The third application example is hot melt coating, or HMC. This process is used, for instance, for taste masking of APIs which exhibit an unpleasant taste, such as acetylcysteine, or ACC. During the HMC process, a suitable emulsifier can be used, in this case polysorbate 65, and this can be added in order to induce crystallization of the stable beta state and to avoid the previously described blooming effect. Furthermore, the emulsifier has a positive effect on the API dissolution rate, and additionally, the HME process provides an improvement of API disintegration into the gastrointestinal fluids. After the process, you will receive particles, ACC particles, coated with DYNASAN 118, and this DYNASAN 118 is now in the beta state and enables smooth and uniform surfaces. If you do not want to use an emulsifier, this is possible, no doubt, but then you have to consider the blooming effect. So if you do not use an emulsifier and you produce these coated particles, directly after the production, you have smooth and unique surfaces. But shortly after production, there will be the blooming effect, and this results in an uneven and rough surface. So in this case, if you do not want to use an emulsifier, you definitely have to apply the previously mentioned curing step in order to get non-blooming and even and unique surfaces of the particles. Let's have a closer look into the process. The hot melt coating is performed using a so-called fluid bed granulator. And this combines a coating partition with a perforated air distribution plate in order to organize a suitable API particle flow. It starts with the ACC particles, which move with the fluidizing air upward, passing a spray nozzle. And this nozzle sprays atomized coating medium, in this case molten DYNASAN 118 plus emulsifier, concurrently with the particle flow. At the end, this leads to uniformly DYNASAN 118 coated ACC particles, or beads as we call it. Now we come to the fourth application example, and this is solid lipid nanoparticles. SLN have been developed as an alternative to emulsions, liposomes, and polymeric nanoparticle systems currently existing. SLN are submicron colloidal carriers which are composed of a lipid, such as DYNASAN 118, which is dispersed in water plus an additional aqueous surfactant solution. The production procedure depends on the desired final particle size, solubility and stability properties of the used API, the drug release profile, and some more. The typical diameter range of SLN is 50 to 200 nanometers. SLN are advantages to use it in process to process purely soluble drugs, because due to its small particle size, the API passes certain cell barriers more easily and can additionally increase the solubility in aqueous fluids, which means at the end, enhancement of bioavailability. What are the advantages of SLN? SLN improve particle stability compared to liquid nanoparticles, for instance. SLN allow to process lipophilic and hydrophilic APIs simultaneously. And with SLN, APIs are embedded high dispersed or molecular dispersed in the lipid matrix. And this means the API is therefore protected against hydrolysis or oxidation. SLN impart at the end more flexibility in the control of the drug liberation profile. And, very important point, SLN can be sterile filtrated. And SLN are suitable for further processing like lyophilization or spray drying. Let's have a closer look into the production process of how SLN are produced. It starts with the materials which are used, suitable lipids, for instance DYNASAN 118 in concentrations of 3 to 10%, and an aqueous solution of a non-ionic emulsifier, in this case Prolonic F68, in concentrations of 1 to 3%. It starts with the first process step, which is preparation of a pre-dispersion. Therefore, the lipid is liquefied, and then a heated water emulsifier solution as well as the API is added, and by applying high shear rates with the use of a high-speed agitator, a pre-dispersion is obtained. It looks like this. And then the next step will happen, and this is the main step, the usage of a so-called microfluidizer equipment. The microfluidizer is a high-pressure homogenizer and reduces the particle size in 3 single steps. In each step, the process is as follows. The pre-dispersion from the first step is conducted through microchannels into the so-called interaction chamber of the microfluidizer. Within this chamber, the material is pushed through a fixed geometry, applying high pressure of approximately 2000 bar via a high-performance pump, which supplies a constant high pressure. During this procedure, very high particle velocities of more than 400 meters per second occur, where the material experiences consistent and enormous shear rates and impact forces. At the end, this results in significant decrease of particle size. This is the main goal, decrease of particle size. So the microfluidizer process acts as follows. The pre-dispersion is subjected to the microfluidizer. This is how the equipment looks like. And then after the first step, you will receive a solution with particle sizes of 130 nanometers. And the liquid of the first step is again subjected to the microfluidizer. And after this process happens, we now have particle sizes down to 95 nanometers. Again, this liquid is subjected to the microfluidizer, and at the end, after the third step, you will receive particle sizes of 72 nanometers. At the end, you have solid lipid nanoparticles loaded with API nanoparticles. So you have the solid lipid nanolayer containing API nanoparticles. So the main application fields of SLN are oral applications such as tablets, pellets and capsules, topical applications like creams, lotions or ointments, and parenteral applications. And this means injections. Generally speaking, if you have any questions which are directed by your customers concerning delivery, pricing, or technical problems, please ask our team, because the whole IOI Pharma team is really glad to assist you. So this is the end of today's webinar. And if you have any questions right now, please feel free to ask.